Skin is the human body's largest organ, weighing some 8 pounds and stretching almost 22 square feet. With skin cancers forming the most common groups of cancers diagnosed worldwide, there is a growing urgency for more insights that could lead to new diagnostics and treatment approaches. Yet among all cancers, there is relatively little understanding of their pathogenesis and how they develop. Being able to study the space-time relationship between cells is changing the game in cancer research. Exactly what Professor Marine's work is focused on.
Melanoma is a very aggressive form of skin cancer, and we believe that it is in part due to the fact that it's a very heterogeneous disease. This disease is composed of many different types of cells. We still don't really understand what these cells do in the tumors and how they contribute to the spreading of the disease. So genomic analysis is a method to analyze the genome of every cell. In a way, is to build identity card of every single cell. And so we are able to, today with these technologies, to discriminate between different cells in the tumors and thereby understand heterogeneity.
Stereosequencing is a method developed by BGI Group that allows us to map the cells in the tissue. With that technique, we can identify every single cell and position these cells in the tissue. That was not possible in the past. It really opens up new possibilities to understand cell-cell communication.
So we've used Stereoseq and a number of other technologies to dissect the intratumoral heterogeneity that one can find in melanoma. This helped us to identify different subpopulations of cells, some that fuel growth, that contribute to the expansion of these tumors, and other subpopulations that fuel metastasis, meaning dissemination or spreading of the disease to different organs. These two subtypes are important because one fuels growth, so by eradicating that population we might be able to decrease tumor burden and tumor growth, and most importantly by understanding and potentially eradicating these cells that disseminate and spread to different organs, these metastatic initiating cells, we might be able to block the spreading of the disease and thereby the dangerous phase of the disease. This is a paradigm shift in the field because we show that this cancer stem cell phenotype can be acquired through reprogramming. This is very important because it shows that tumor growth can be stimulated by almost any cells in the tumor if they are in close contact to blood vessels. Understanding that, of course, allows us to potentially develop therapeutics that target this communication between endothelial cells and cancer cells and thereby block tumor growth.
Working with researchers like Professor Chris Marine and oncology experts like Professor Bechter is what drives BGI Group to develop more innovative ways to map the world's DNA to help us better understand the basic building blocks of life.
What we have learned in the past years is that the complexity of the tumor is so big that you need to focus down, you need to boil down to really cell-cell interactions, and you need to understand which cells communicate with each other. And only if you decipher this communication pathway, you will be able to intervene with a treatment strategy. The more and the better we understand the heterogeneity within the tumor, the more we will be able to delineate new treatment strategies which actually aim at the heterogeneity which is present in a given tumor. So we will be able to, tailor treatment strategies more based on the knowledge we have from genomics.
So using the BGI technology, we've been able to identify and map important subpopulations of melanoma cells in the tissue. These technologies really teach us a lot about how these tumors grow, how these tumors spread, and I think we've learned quite a big deal already, but this is just the beginning. These technologies will really allow us to better understand these processes and eventually how to interact and intervene therapeutically.
Okay, well, hello everyone. Good afternoon or good morning, wherever you are. Well, welcome everyone joining the webinar today. My name is Tibi Duan. I'm the Product General Manager for Stomix Global. It's a very honor for me to be here today and to introduce the Stereoseq OMNI, our very new product solution, focusing on discovering spatial transcriptomics information for FFPE samples. So before I dive more into our new product solution and about StereoSeq, one question that continuously bring up by our users and our potential customers is why is it needed for them to use spatial technology? So spatial transcriptomics technology emerges as a new technology that emphasizes on obtaining omics information in a spatial context. And compared to single-cell RNA sequencing, which has been successfully adapted and utilized in many applications, spatial transcriptomics can identify the position of a cell in relation to its neighbors and surrounding structures, which can provide valuable insight into the cell's phenotype, state, and ultimately its function within the tissue. So established in 2020, StoMix has been offering a cutting-edge spatial temporal multi-omics platform that help researchers to find unbiased answers to biological questions in both scientific research and clinical applications. So StereoSeq, spatial enhanced resolution omics sequencing is the only spatial technology currently on the market that can access the whole transcriptome with single cell resolution, at the same time achieve a subcellular data with a 500 nanometer resolution. So StereoSeq has demonstrated its promise in simultaneously profiling hundreds to thousands of genes at subcellular resolution, and this is important for a more comprehensive analysis. In one of the earlier studies, scientists were able to study the kinetics and the directionality of transcriptional variation in the time course of mouse organogenesis using StereoSeq. But organogenesis and ontogenesis are not the only applications that StereoSeq can help you explore. There are many application scenarios for StereoSeq, particularly crucial for spatial mapping of complex tissues such as the brain, embryo, tumor samples, and also restructuring the cell developmental pathways by identifying the differentiation state of the cells, and also for immune cell profiling for immuno-oncology research, specifically with the involvement of tumor microenvironment. And additionally, also spatial computational biology by combining RNA spatial data with imaging data to identify cell morphology for a more accurate cell segmentation and spatially resolved single cell gene expression study. So all of these were able to achieve with StereoSeq's pattern DNB arrays. So the ability of StereoSeq's high resolution and large field of view is empowered by this DNB-seq features. So DNB stands for DNA nanoballs, which is the foundation for StereoSeq. And our standardized DNB chips have spots with approximately 220 nanometer in diameter and a center-to-center distance of 500 nanometers, which can provide up to 400 spots for tissue RNA capture per 100 micrometer square. So for the DNB templates are contains the random barcodes, and they are deposited on the pattern array, incubated with primers, and then sequenced to obtain the coordinate ID. So this workflow, StereoSeq were able to achieve the tissue-to-data solution for fresh frozen samples. However, fresh frozen tissues, even though it's the gold standard biospecimen type for NGS-based spatial biology study, collecting fresh frozen samples is usually not feasible because clinical works deliver formalin-fixed paraffin-embedded tissue blocks. So now with StereoSeq OMNI, we are trying to address this question by bringing a revolutionary sequencing-based spatial transcriptomic solution to study the FFPE samples, and in order to obtain more insightful pathological knowledge with higher resolution. So StereoSeq OMNI is able to achieve a true single-cell level gene expression profiling combined with the histology information. Because we're utilizing an innovative chemistry design for our probe, we're able to capture the total RNA within any species. So it's species agnostic, and as well as studying microbiome on RNAs and also the host on the microorganisms interactions in a spatial context. So for Stereoseq OMNI, we're specifically designed and skillfully engineered in order to capture degraded RNAs within FFPE samples, and as well as open new avenues to study your FFPE samples that has been stored for previous applications. For the principles of Stereoseq OMNI is still utilizing the DNB patterned arrays. But we're replacing the capturing primer with a random probe that can efficiently capture degraded RNAs within FFPE samples, as well as capturing total RNAs, including mRNAs, microRNAs, long non-coding RNAs, for further exploration. And for the DNB pattern arrays, we're able to achieve various sizes of the resolution in bin sizes. So for bin 1, which is 1 DNB resolution, and typically a bin 20 is a equivalent size to a cell. But at the same time, because we're incorporating staining methods into Stereoseq OMNI and also conventional Stereoseq workflows, we're able to achieve a single cell analysis utilizing nuclei image assisted cell binning methodology. So this is a general workflow for Stereoseq OMNI. So the typical categories will be performing the Stereoseq FFPE transcriptomics experiment, and then perform sequencing, obtain the sequencing data, and then utilize the Stereoseq softwares and also proprietary algorithm pipelines to obtain the spatially resolved gene expression profile. So with the FFPE tissue blocks, we'll first mount it onto the Stereoseq chip slide, and then perform a series of biochemistry workflow, and then also incorporate staining and microscopic image into the workflow, and then eventually capture the RNA, and then perform in situ cDNA synthesis to obtain the library. And then we'll utilize the DNB-seq platforms for sequencing of the libraries. And with Stereoseq OMNI, it's important to look at the overlays of the histology information with the high resolution spatial transcriptomics. So for Stereoseq OMNI, we're incorporating H&E staining to better study the histology and also the morphology of the different types of tissue. But at the same time, it's also available for staining the nuclei if the spatial single cell analysis are important for your research. And we also provide these visualization softwares that are able to for you to overlay the image, the tissue mask, and also the single cell expression mask files onto the within the software, be able to look at the cell cluster results, and also using third-party algorithms, obtaining the cell annotation results. Our software can also take in those data and then display it using this visualization platform called StereoMap. And besides that, because we're utilizing the random probe approach, and it's easier for researchers to look at the long non-coding RNAs and also other RNAs in a spatial context, and to look at the distribution and to infer further how they're affecting the cellular function in disease and healthy states. So this is a breast cancer tissue that we're utilizing to look at the cell typing and also to infer some of the cell annotation results and their clustering results at a very fine resolution using Stereoseq OMNI. And here I wanted to demonstrate one of the demos that we were able to do is to characterize the spatial gene expression profile on breast cancer FFPE samples. So we're looking at the dysplasia versus the tumor regions. In dysplasia, the cells might look abnormal under a microscope, but they are not yet at the cancer stage. And a lot of information can be told when staining the section with H&E staining and when looking at them under the microscopes. So this histological information is useful for researchers to select tissue regions of interest related to specific scientific questions for exploring further potential molecular mechanisms, especially for FFPE samples, which contains a lot of information that can be further studied past the conventional imaging studies. So some studies have found that the patients with biopsy-proven benign mammary dysplasia have a higher incidence of breast cancer than those without it. And with stereoseq OMNI, we were demonstrating that there are differences in the spatial molecular characteristics between dysplasia region and the tumor region, from tissue level to cell level in breast cancer tissues. So we're able to see that there are more cancer-associated fibroblasts, CAFs, and epithelial cells observed in the dysplasia region than in the tumor regions. And since CAFs secreted cytokines involved in the recruitment and maturation of macrophages and other immune-related cells, so they tend to increase the availability of the tumor to immune cells and promote anti-tumor immune responses. And we do observe that kind of behavior, and also when we look closely at the immune cell type compositions, and also when we look at the pseudo-bulk differential expression analysis, and we did observe that the region of dysplasia had higher immune cell filtration, such as T cells, B cells, and perivascular-like cells, which is a type of cancer-associated fibroblast. And also further analysis at the molecular level also revealed that the region of dysplasia was also enriched with significant immune responses, such as MAPK signaling pathway, which is essential in regulating many cellular processes, including inflammation, cell stress response, and also cell apoptosis. So one of the applications that were demonstrated using stereoseq OMNI, this is one of the earlier works that were done prior to we were preparing for the official workflow that will be launched in July. We're lucky to have researchers from MD Anderson to be involved in our early access program last year. And they truly demonstrated the ability for stereoseq to obtain to actually discover unfound markers that has not been previously published in articles related to ovarian cancers, and also the ability for stereoseq data to merge with other platforms to really achieve multi-omics and multi-modality study in when studying the 3D tumor microenvironment. And they also describe in the paper that omics methods, the stereoseq OMNI holds the highest resolution so far of any non-targeted assay by at least an order of magnitude. And in this study, they were looking at high-grade serous ovarian cancers. So the high levels of tumor heterogeneity were studied using stereoseq analysis, combining with UMAP and the latent clustering analysis. So in this study, they've identified a collection of markers. So the one on the top left here is the spatial gene expression obtained from stereoseq OMNI, and they also incorporated other multiplex seq-IF method to obtain this tissue segmentation, identifying the tumor area, the stromal area, and also the interface area in between these two regions. And they utilize the UMAP dimensionality reduction and be able to look at specific markers within the tumor and the stromal region. Some of the markers related to ovarian cancer, for example, the MUC16 and the WFDC2, and also you can see a pattern of them co-localizing with the tumor areas, as well as some of the stromal markers, the collagen alpha 1, and also the IGF BP7 tend to co-localize with the stromal areas as well. And so they're only showing one of the layers that are being studied, but some of the ones interesting marker that they identified, which is because stereoseq OMNI were able to capture the long non-coding RNA, which is AL35 07.1. This has been shown to be associated with advanced stage clear cell renal cell carcinoma and osteosarcoma metastasis, and which was found to be highly expressed in a particular cluster of cancer cells in the ovarian cancer tissue that they were studying, which was has not been studied before in a spatial context. And they were also able to combine omics data with mass spectrometry imaging and also multiplex-seq IF with the stereo-seq OMNI data to create multi-layers of information to actually reconstruct the 3D molecular information of this tumor microenvironment. So for those who are interested in this paper, please go check it out. It's a very interesting work. And since we're launching the product in July, we are now offering the early bird promotion for users who are interested in using the stereo-seq OMNI workflow. Currently, the early bird promotion period is between April 11th to June 28th. So if you're interested and wanted to get to know more about stereo-seq OMNI applications and what can be utilized on your platform and within your project, please do contact us and let us know how we can be of help.